So thank you for the invitation to come and present to you all today. I really appreciate being invited by the students. Several of your colleagues have attended the Kattner Institute, and that's how I met them and started interacting with them. So I'm really glad to be able to come and present a little bit about what my lab studies. What I've shown you on the first slide is a couple of important characteristics of Neisseria gonorrhea. This slide here is typical of the clinical presentation of gonorrhea, particularly in a symptomatic urethritis caused in males. The organism actually gets into polymorphonuclear leukocytes and it hides out there, and it can grow quite well in PMNs. Its very close cousin, Neisseria meningitis, does exactly the same thing, but it gets into the PMNs in the CNS, in the cerebrospinal fluid, and causes bacterial meningitis. So this is a very typical clinical presentation for the pathogenic Neisseria. What I've shown over here is a schematic which we published recently. It was actually a cover image for Molecular Microbiology describing this iron acquisition system, which I'm going to spend most of my time on today. We're not going to talk about all virulence factors. Sure, yeah. We're not going to talk about all virulence factors in the pathogenic Neisseria. There are lots of them, but I'm going to focus primarily on iron acquisition. So why Neisseria gonorrhea? What's important and what I hope you will take away from this presentation is why this bug is re-emerging as an important pathogen. It's a gram-negative organism, and it's an obligate human pathogen. It doesn't infect any other animals. It's exclusive to humans. It's a reportable disease, and there are more than 300,000 cases reported in the United States every year. It's been estimated to be the most common bacterial infection in the world. These are reported cases. The real number of cases is probably at least twice that. And antibiotic resistance is a huge problem with the gonococcus. This next slide shows the dramatic increase in resistance to what was the go-to drug to treat gonococcal infections, ciprofloxacin. And by 2007, 2008, almost 20% of isolates that were screened were resistant to that go-to drug. So in 2008, the CDC said you can't use that as the drug of choice. So now penicillin's gone, tetracycline's gone, the quinolones are gone, and we're basically left with a single class of drug to treat these infections. And that was ciprofloxacin. That was, the third generation cephalosporins. But unfortunately, as you've probably seen in the lay press now, resistance to this drug has emerged as well. So we're really looking at a superbug status for this organism. A new strain is resistant not only to the earlier drugs that we use, but also to this last line of effective drugs. And so there's the concern that we'll soon be in a situation where gonococcal infections are untreatable. So there's also been this long understood, or well known at least, correlation between HIV transmission and gonococcal infections. So people who have both infections simultaneously have higher HIV titers in their secretions. And if you treat the gonococcal infection with antibiotics, then the HIV titers go down. So there's some kind of synergistic relationship between the two. And so there's a hope that if we could prevent the bacterial STD, that we might actually decrease HIV transmission as a consequence. There's no vaccine to protect against gonococcal infections, and that's in large part due to a hugely variable surface structure. Virtually every antigen they put on their cell surface varies at high frequency. Their LOS changes at high frequency, or their LPS. Almost all of their surface antigens change at a rate of 1 in 100 to 1 in 1,000 cells. So it's a completely moving target. Also, an infection is not protected, so people who get infected are not protected against a subsequent infection. And people do get infected over and over and over again. And so one of our main chief goals is to identify more stable targets that we could use as potential vaccine candidates. So what would you like to see in an ideal vaccine candidate? Well, it needs conserved from one strain to another. It needs to be surface exposed. It needs to be expressed in vivo in the host, in the human. And it needs to be critical for growth in vivo. And so we focused on iron acquisition pathways as potential vaccine antigens because they fit all of these categories. Some of them are very well conserved. They are surface exposed. They bind to the same ligand. They're expressed in vivo, and they're necessary for survival. And we focused most of, well, here's a slide that explains how the Neisseria go about getting iron from their host. It turns out humans have sort of gotten on to the program, and we understand, our systems understand, that iron is an important nutrient for most bacteria. And humans actually go to great lengths to hide iron away from invading pathogens, and not just bacterial pathogens, but fungal pathogens as well. And even some viruses need iron as well. So there's this ongoing battle between the host and the pathogen for iron. And most bacteria overcome this limitation by secreting low molecular weight iron chelators called siderophores. And they're like little iron cages, and they solubilize the iron and bring it back to the bacterium. But the Neisseria don't bother with that. They don't send out any siderophores. They just hijack our iron. They can use our transferrin. They're exquisitely attuned to human transferrin. They can't see any other mammalian form of transferrin. Likewise for lactoferrin and also for hemoglobin. So they can use all of these iron sources in a specific receptor-mediated event. Just to remind you, transferrin is found in the serum, and it's a molecule that ferries iron around from the liver where it's made to other places where iron is required. Lactoferrin is found primarily on mucosal surfaces, and hemoglobin is found inside of red blood cells. The bacteria can use some of these siderophores that are produced by other bacteria. So they don't make anything themselves. They just hijack it from their host. And in fact, we termed this process iron piracy some time ago. So we focused, and on the previous slide I highlighted in red, we focused on transferrin iron acquisition for a couple of reasons. And that is that all gonococci can use human transferrin as an iron source. The receptor, as I said, is exquisitely sensitive and only recognizes human transferrin. In the process of iron internalization, it's only the iron that comes into the bacterial cell. It's not the whole transferrin molecule, which is 80 kilodaltons, stays on the surface. So there's a stripping process that has to happen at the cell, at the bacterial surface. And there are two proteins that are involved in this process, and I'm going to give you a lot of molecular detail about these two proteins. They both bind to transferrin individually, independent of one another. There's a transferrin binding protein A, which is an integral membrane protein, and I'm going to show you now the known structure of that protein in just a minute. And the TPP-B protein is a lipoprotein. It's N-terminally modified with palmitate, and that allows it to be tethered to the outer membrane of this gram-negative organism. And both proteins are specifically expressed under conditions of iron depletion. So when iron is low, this system is turned off as a means to acquire. So when I was a postdoctoral fellow at UNC Chapel Hill, we tested whether a mutant of the gonococcus that couldn't make this transferrin iron acquisition system was virulent. And we tested that in a human infection model. So at UNC, this is the only place in the whole world where this human infection model exists. And what we did, and what is continuing to be done in this model, is that human males, volunteers, are inoculated intraurethrially with the gonococcus. And so we were testing the wild-type strain in comparison to a mutant that could not make these transferrin binding proteins. And we asked, was there a difference in their ability to cause infection? And so the controls are shown first. In two different trials, we had 3 different subjects which were inoculated with the wild-type strain, and urethral discharge was observed either at day 3 or day 4. All of the swab specimens, which are collected for 5 or 6 days, all the swab specimens were positive, and all the urine specimens were positive. So that's our control. We know these organisms are infectious. By contrast, the mutant that could not express either of these transferrin binding proteins was not able to cause any urethral discharge at all in any of the 7 subjects. The organisms were never recovered in the swab specimens. There was one person who had a transient bacteriuria, but it was resolved spontaneously, and the rest of the urine specimens were negative. So we concluded from this that a mutant that can't make this system is exquisitely attenuated in the human male and can't even initiate an infection in the human male. We can't do this experiment in women ethically because the infection